Hello everyone, welcome to our design day video. Our project is about wastewater treatment process and our project number is 21084. My name is Karim Alsanan and I'm chemical engineer.
I'm Elijah Rollison. I'm a chemical engineering.
I'm Jared Dutton. I'm also a chemical engineering major.
I'm Nervous Rina, also a chemical engineering major.
I'm Mohammed Al-Aish. I'm also a chemical engineer.
Our mentor is Dr. Sang Tao and Mohammed will start with the introduction.
Okay, the goal of this project is to build a process that treats three different types of wastewater: mining, agriculture, and industrial wastewater. This project will be used to teach undergraduate students majoring in chemical engineering at the University of Arizona about wastewater treatment processes. The process flow rate of the whole system need to be adjusted at 2 gallons per minute. This project is shared with another team from the University of Arizona to design the process and install the equipment. The design has three paths, and each type of waste will go through its path. The treatment system will include unit operations such as reverse osmosis, coagulation, flocculation, UV light treatment, and membrane bioreactors. Our team focused on designing the reverse osmosis and membrane bioreactors. The final product of this project will comply with the EPA and Arizona regulation of potable water. Also, all the wastewater generated by the wastewater treatment lab must be collected and disposal as per EPA standards.
In general, wastewater treatment is a multiple barrier sequence of processes used to remove contaminants from wastewater through physical, chemical, and biological processes. The treatment should be able to convert the wastewater into an effluent that can be safely returned to the water cycle with adequate impact to the environment. Usually, a large-scale wastewater treatment plant involves a primary, secondary, and tertiary treatment. Primary treatment focuses on suspended and large solid removal by the physical process of screening, grit removal, and sedimentation. Meanwhile, secondary treatment includes biological processes to remove nutrients and organic compounds via microbial activity. Finally, tertiary treatment removes the remaining inorganic contaminants, heavy metals, and excess chemicals so that the final effluent is free from all the contaminants before being transported back to the environment. And we have three different type of wastewater, and we have the industrial, and it's mainly containing inorganics and suspended solids. And we have also the agriculture, and it's about organics. And lastly, we have the mining wastewater, which about it's heavy metals.
Sorry. Reverse osmosis is the process wherein wastewater flows through a semi-permeable membrane and removing the contaminants. In osmosis, typically solvent moves from areas of low concentration to higher concentration. However, using the high pressure, we can overcome the osmotic pressure, therefore reversing normal osmosis. Thus, it's called reverse osmosis. Reverse osmosis can have rejection rates as high as 99% of certain contaminants. In the experiment with the RO we have designed, students will be comparing rejection percentage with the operating pressure. As an example, here we have the operation pressure with the calcium rejection rate, and as well as a PFD of the reverse osmosis. Next, we have Nerd talking about the MBR.
Okay, membrane bioreactor is the combination of a biological wastewater treatment process with a membrane process like microfiltration. The reason why MBR is now widely used for industrial and municipal wastewater treatment is because it has a smaller footprint compared to other conventional process. This project focused on designing a lab-scale submerged MBR for future chemical engineering students, and this MBR should be capable of achieving a high-quality effluent with more than 90% removal of BOD5, total nitrogen, COD, and so on, as shown in the table below. Submerged MBR is our preferred configuration due to its low energy consumption, high biodegradation efficiency compared to side-stream membrane bioreactor. There are factors that influence filtration in MBR, for example, the membrane characteristics, sludge characteristics, as well as operating condition. In the future lab, students should be able to experiment with these variables and analyze how a control variable can affect the system performance. Okay, moving on to the PNID of membrane bioreactor. The wastewater treatment using an MBR starts once the effluents enter the feed stream and flow into the reactor. Since our design focus on the submerged MBR, the hollow fiber membranes will be assembled and installed into the reactor. Air compressor is included to operate the systems, and the process blower is crucial in this design for oxygen transfer to the reactor. The reactor tank is where the biological process is happening, and once the water is pulled through the membrane, it will be pumped out to the permeate tank or to the next treatment process. Excess sludge will be disposed properly according to the sludge disposal regulations. Fouling and fouling control are our major considerations when designing an MBR, since membrane fouling is the most serious problem affecting system performance. Air scoring around the membrane and a weekly cleaning using citric acid is very helpful to remove or at least reduce the fouling layer. Next, Jared will talk about the lab's experiments.
As one of our objectives is to help students better understand the processes behind wastewater engineering, we want them to have a hands-on experience with the equipment involved in the system we've developed. This can be accomplished in the form of lab experiments for both RO and MBR units. With the RO unit, students will attempt to understand the importance of pressure on the purity of the permeate stream by sampling it at 3 different pressures between 150 and 400 psi gauge. As for the MBR unit, students will be controlling the temperature inside the bioreactor tank to observe the effects that temperature has on nitrification, COD, and nitrogen removal from the permeate stream. By taking samples from these streams, TAs will use a spectrophotometer to reveal the concentrations of the remaining contaminants inside the product stream for the students to analyze. Outside of lab time, they will be asked to form calculations with their data, interpret the trends with their results have created, and perform error analysis to assess the validity of their data. Upon completing these experiments, we hope that students will become more comfortable operating these pieces of equipment for the future and receive valuable learning experiences that might not be offered in a classroom.